Okay. A little bit loud, but okay. Okay, so before I start, did you have any questions that you wanted to ask? I think I've got everything that I need. That's fine. I've had a couple people that were kind of delayed getting started, and so I always advise to people go ahead and at least put something up on the discussion board and go ahead and get your assignment turned in. Um, you might not be able to get full credit for like the discussion for not posting early in the week and that kind of thing, but at least you can get um, almost full credit. You can get some points for that, and so that the points all add up at the end. So the more points you can get, the better off you'll be, and that way you can get some points out of it instead of not having any points. And the and I'll go back to the discussion board and see what additional things have been posted. I've already gone through and graded the discussion board, and I'll probably go through graded the first discussion board, Module 1 discussion. And I'll probably go through today and grade the Module 2 discussion, but I can always go back and see who has participated that doesn't have a grade, and I can go in and get some points for people. So it's always a good idea to go ahead and post something, even if you're running a little bit late on that. So I had replied to somebody in email and kind of let them know the same thing. So that's a good question for some people that are still trying to get caught up. It's always a good thing to go ahead and go through the exercise, because that's part of what I'm looking for, is that you've kind of done some research on the topic and you're kind of making those connections. Ideally, you'll do that in the context of the discussion, but if you're not able to do that, if you've missed the discussion, you can still get some points for the work that you've done. That was kind of a rambling answer to your question. But I'd had a couple people ask me that also, so I responded in email to a couple people. So what I want to kind of go through today is going to hit some of the aspects of all three of the chapters that we're I've had a couple people talk to me or express some concern about the fast-paced nature of the class and how there seems to be a lot of content for each of the weeks. And that's always a concern in a summer class. When you take a 16-week semester and you compress that into a 7 or 8 week summer session, you are already working at double the pace that you would be. So, for example, while we might take a whole week to work on the skeletal and muscular system and a whole week to work on the nervous system, we're having to compress that into 1 week in the summer. And the other issue is that it's an online class. It's not a face-to-face class. So, if you were taking the class face-to-face, you would be spending 6 hours a week in class in the classroom. And then you'd be spending another 12 to 14 hours a week outside of class. Because the rule of thumb that we usually tell students is at least 2 hours a week outside of class is what you need to spend working on learning the material for every hour that you spend inside of class. So it's a lot to take on a summer class, and then when a lot of that is going to be independent learning in an online class, then that does make it seem to be accelerated and a lot of material. And for some people, that can be overwhelming. And that's one of the reasons we don't always recommend a summer class in this accelerated format to all students, because not everybody has got, the resources that they need to be able to work on that. So I am aware that it's a lot of material, but if you're going to get a credit for the class, you have to have covered the same material, whether you take it in a 4-week or a 6-week or 8-week or a 10-week or a 12-week or a 15-week semester. So I am trying to be sensitive to that. There's a lot in Chapter 13 that we're not covering this week. So I do know that it's a lot of material, and that's one of the reasons why I wanted to do these kind of Wimba lectures, would be to kind of do some explanation for some of the things that people found confusing, and hopefully get some questions answered. So I have this slide in the PowerPoint, which is kind of an overview, and it kind of is a justification for why I put these 3 chapters together. 5 and 6 are the next 2 chapters in the book, the skeletal system and the muscular system, but then the nervous system isn't until chapter 13. And so I thought people might be wondering a little bit why that was thrown into this. And I think you'll see, and especially when you work on your discussion board prompt for this week, you'll see that really I think the muscular system and the nervous system fit more closely together than the muscular system and the skeletal system, as far as the pieces that have to work together for it to function. Now, the skeletal system and the muscular system work together very closely functionally because muscles move bones. And that's how our body moves, is the muscles move our bones that move our body parts. But then to control all of that uses the nervous system, and so that's kind of why I threw that in. Now, I'm not going to go through a whole lot from Chapter 5. Chapter 5 is on the skeletal system, on bones, and that's like skin. It's a pretty simple type of a system. It's a good one to kind of start with because there's not a lot other than learning the names of the bones and the locations of the bones. There's not too much that goes along with the physiology of the skeletal system. So I just want to point out a couple of slides from that chapter. This particular slide shows you some terms. Again, with all of these chapters, there's going to be some terms and terminology. So whatever is working for you as far as doing note cards or doing charts or writing it out on a sheet of paper, whatever is, doing sticky notes on your mirror so you can see the words. Had somebody tell me that worked for them really well. So whatever your method is for learning some of the terms, there's some new terms in this chapter to look at. There are 3 types of cells that we talk about when we talk about bones. The prefix osteo lets you know that you're probably talking about bones or something related to the skeletal system. And so mature bone cells are called osteocytes. That literally means bone cells. Cyte meaning cell, osteo meaning bone. So these are the mature bone cells that live in the bone matrix. Previously, they were a type of cell called osteoblasts. Osteoblasts help build bone. They secrete collagen and other proteins, and they secrete that matrix that becomes calcified to be bone. And then once the bone is built, they differentiate into or become osteocytes that live in the bone and help to remodel the bone. And then osteoclasts are cells that break down bone. They secrete acids and enzymes to help break down bone. Bone is a major store of the mineral calcium as well as magnesium and phosphorus. Calcium is required for a number of cellular functions. Calcium is required for muscles to contract. Calcium is required for nerves to signal. We're going to see that in less than an hour. So the body has to maintain the level of calcium in the bloodstream, and that's sometimes referred to as calcium homeostasis. And these cells are involved in that process. Osteoblasts can help build up bones. So if we have an excess of calcium in the bloodstream, those cells can put that calcium into bone to store it. And then osteoclasts can break down bone to get the calcium back out. So if the levels of calcium in the bloodstream are too low, that's the homeostatic mechanism that we use to regulate blood calcium levels. And so those are some cells that are involved in that process. The basic unit of compact bone is something called the osteon. The osteon is kind of this unit right here. It's a round unit that's made up of concentric layers of bone tissue. This is actually what it would look like under the light microscope if you looked at it under the microscope after the tissue had been stained. And then part B shows you kind of an artist's rendition of what the bone looks like, showing you lots of these different structures. And so they show you the osteon. Through the center of an osteon runs a blood vessel in a space called a haversian canal or the central canal. This osteon is often called a haversian system or a haversian structure. That's capitalized, so that means it's named after someone. So there was some guy who named this after himself when he discovered it. There are also blood vessels that run sideways through these sideways canals, and those are called perforating canals. They are also called Volkmann's canals. Again, that's capitalized, so it's named after somebody whose name was Volkmann, who discovered them, who named it after himself. Now, the really small dots that you can see in this figure, all of these little bitty dots that are all throughout here, those are the cells. Those are the osteocytes. And the osteocytes sit in spaces called lacunae, which is Latin for little lakes. And the lacunae are connected by small spaces called canaliculi, which is Latin for little channels. And so these terms are in your text that's near where that figure is. Page 88 is where you'll see canaliculi. It says these little canals allow nutrients to move through the bone matrix. So the bone matrix is very solid. In order for nutrients to diffuse to those cells, it has to go through these spaces called canaliculi. And if you look at the photomicrograph, you see these little bitty hair-like structures. Looks like strands of a spider web. And those are the canaliculi that you can see. And so in this osteon, in this round piece of bone, there's one blood vessel in the center. And that's bringing nutrients to all the cells that are in all those layers. And so the nutrients and the gases will diffuse outward from that central canal, from that blood vessel, through those canaliculi to get to all of those cells. And so this is this is a slide that gives you some of those terms as well. So central canals that run through the bone that have the blood vessels in them. Around those central canals are these layers, these concentric layers, or lamellae. Lamellae means layers. There's these rings of this bone matrix, and it's very similar to the way a tree grows, rings of a tree trunk. The spaces are called lacunae, and that's where the cells sit. And the canaliculi have the extracellular fluid, and that's what allows diffusion of nutrients. And that diffusion goes outward from that blood vessel that's in that central canal. Or if it's waste, they diffuse toward the blood vessel so the blood can carry them away out of the bone. And so that's some of the things microscopically to look at about the bone tissue. Now, in addition to looking at the bone on a microscopic level, we can look at the bones on a macroscopic level. And this is a figure from your book that shows you the names and locations of a lot of the bones that are in the body. You'll have some questions on your midterm that will ask you about the terms for some of the bones, and you'll actually need to fill in some of the terms for the bones. I don't accept common terms for identification of the bones. So if you see a picture of a bone and you think it's the shoulder blade, you will need to say scapula because that's the appropriate term for shoulder blade. Find that right here. If you see a bone and you think it's the patella, the kneecap, you'll need to say patella and not kneecap. So you'll need to type in the you will need to not use the common names for the bone. You will need to use anatomically correct names for the bones. In an in-class type of a situation, I usually give students just kind of a blank skeleton to fill out so that they can start to learn the location and the names of the bones, and spelling will count. Given that this is going to be similar to an open book test, since it's an online test, even though it's timed, what I'll probably do is show you photographs of bones and have you identify the bones that way. That's more similar to what we do in the lab type course. And obviously you can look up the name of the bone, so spelling will count. It will be marked wrong because it'll be automatically graded. It will be marked wrong if it's spelled incorrectly. And so that's another reason that you're going to need to use the correct term, like clavicle for collarbone. Since it's automatically graded, it won't recognize if you put collarbone. It'll only recognize clavicle. So this figure is going to be kind of helpful to you to start to learn some of the names of the bones and some of the their locations where they are. And it's always fun to impress your friends when you know names of bones that they don't know. So now from here, your chapter starts to talk a lot about joints. And I'm not going to get too much into joints. There's a lot of more detailed things that we can learn about the structure of joints and synovial joints and those kinds of things. But I do think that knowing some of the terms related to joint movements is helpful. And so your book gives you this figure 510, that's in your book on page 97. It's a little bit easier to look at in the book. And this gives you some terms and examples of what the terms mean. So when we're talking about extension, when you extend a part at a joint, you're actually increasing the angle between two bones. And then when you flex, you're reducing the angle between the two bones. So when you flex your elbow and move your lower arm towards your upper arm, you're decreasing the angle between your lower arm and your upper arm. And so they show you some example of flexion and extension at the knee and the elbow. And then movement sideways is going to be abduction if you're moving it away from the body, or adduction. And so they show you some examples of that. Circumduction is moving in a circle, where rotation just moves a body part around its axis. They show you supination and pronation. That tends to be more specifically related just to the bones of the forearm. And then they're showing you gliding movement that can occur at the wrist. You can have gliding, sideways movement that happens at the wrist. So you might want to take a look at the terms on this page because there probably will be some matching questions on the midterm related to movement at the joints. So then I want to kind of spend the bulk of the time talking about the chapter on muscle and the chapter on the nervous system as it relates to muscle contraction. That's probably going to be the bulk of what I want to go over, because that's the part that is going to be more difficult for you to do independently. The rest of the PowerPoint is more straightforward. You can read and go over the PowerPoint and look at the animations and kind of get an understanding of that. But the muscle contraction piece is the piece that's going to be more difficult to do independently. And so that's the part that I want to try to explain. And your book gives it to you in pieces, kind of in layers, and kind of layers on more and more information. And I think that's a good way to do it, because trying to go through it from beginning to end and learn all of the detail of it from beginning to end is going to be really overwhelming. So by breaking it into pieces and then putting those pieces back together, we can kind of bite off chunks that are easy to explain, and then try to put the whole thing back together. So because I was a few minutes late starting this morning, I probably am going to run over an hour. I'm probably going to run over the 11:30 mark. So I'm just going to go ahead and continue to record until I go over all the pieces that I want to go over. But I wanted to let you know that, Sarah, I won't be offended if you have to leave, if you can't stay for the whole thing. So if you needed to leave before I was finished, I just wanted to let you know that I'll probably go over today, because I want to make sure and hit all of the stuff that's about muscle and the action potential in the nerve channel chapter. So we're going to spend the bulk of our time today talking about skeletal muscle, but there are three types of muscle, and they're all very similar. So we can use skeletal muscle as a model because it's the more complicated one, and we can learn the details about it. But the way that skeletal muscle contracts applies to smooth muscle, and it also applies to cardiac muscle. Smooth muscle cells surround some of the hollow organs, like your intestine, and help to contract to propel food through the intestine. And so smooth muscle is under involuntary control. You don't think to yourself, okay, now I need to contract the muscles around my intestine. That just happens as a normal part of the digestive process. Cardiac muscle is also involuntary. You don't think to yourself, okay, I need to contract the muscle that pumps the blood through my body. So smooth muscle and cardiac muscle are both considered to be involuntary muscle. Involuntary. And then skeletal muscle is what we call voluntary, because it's under nervous system control, and you can usually make the decision, okay, the nervous system is going to control this muscle. Now, sometimes it's not under conscious control. Sometimes your muscles contract, for example, when you shiver. Sometimes some people have involuntary skeletal muscle contraction, but for the most part, we can make the division between voluntary and involuntary, as the skeletal muscle is voluntary and smooth and cardiac muscle are types of involuntary muscle. So we're going to talk first a little bit about how muscle tissue is organized, what the terms are for the parts of muscle tissue. And we'll go all the way from a muscle down to its microscopic parts like protein filaments. Then the next thing that your book talks about is the actual mechanism of how muscle contracts. And that's that protein filaments slide relative to one another, and that's what contracts the muscle. And so that's often called the sliding filament mechanism of muscle contraction. And so we go straight to that microscopic mechanism of how muscle contracts. Then your book has a section on calcium and how calcium controls muscle contraction. So that'll be the next piece that we add. Once we know about those sliding filaments, we can look at how calcium relates to the sliding filaments. And then after we look at calcium, we can answer the question, what causes calcium to be released? And then I made a little bit of a mistake on this slide. I was obviously typing too fast. Then we'll look at how a nerve triggers a muscle to contract. And that will lead us very directly into our discussion in Chapter 13 on the details of the nervous system and how a nerve cell works to trigger a muscle cell to contract. So that's kind of an overview, and hopefully that'll help you kind of fit together some of the pieces that we're talking about. And then at the end, we're going to go through the whole process as a whole and try to fit all those pieces together. So this is a figure that lists the subdivisions of skeletal muscle from the largest to the smallest. And so we start out with a muscle. And a muscle is comprised of groups called fascicles. The fascicles are made up of muscle fibers, and that's the level of the cell. So those are muscle cells. Muscle fibers are muscle cells. They're made up of small organelles called myofibrils. And the myofibrils are made up of protein filaments. And so this is a figure from your book that shows this whole large muscle. Then it shows these fascicles, which are these bundles of muscle cells. Each has its own connective tissue sheath. So each is surrounded by a piece of connective tissue. And so this muscle fascicle is made up of these muscle fibers. And this muscle fiber is made up of an organelle called a myofibril. So if you put a bunch of myofibrils together, you get a muscle cell. If you put a bunch of muscle cells together, you get a muscle fascicle. And if you put a bunch of muscle fascicles together, then you get a muscle. And your book gives you this figure also that's on page 108. And it's a little bit easier to see if you look at it in your book. But I wanted to show you the whole figure here. So part A is just what we looked at, is the muscle being kind of broken down or subdivided down into a myofibril inside a muscle fiber. And then in part B, they show you what this looks like under a microscope. And you can see that there's lots of little lines that you can see when you look at it under the microscope. So in part C, they kind of show you an artist's rendition of what you're looking at here so that they can kind of give you a better idea of what it is that you're looking at in this microscope picture. And they show you an even further blow up of the microscope picture. So we're looking down at the smallest unit of muscle, which is something called the sarcomere. One myofibril that they're showing you one piece of a myofibril that they're showing you in this artist's drawing is made up of sarcomeres. And the sarcomeres are made up of protein filaments that we call thin filaments and thick filaments. Part D and Part E show you in a little more detail the thin filaments and the thick filaments. The thin filaments are also called actin filaments because they're made up of a protein called actin. Actin is the subunit, and it's put together in this long kind of helix to make this long filamentous protein. There is another protein that's part of this thin filament. They show you these long green lines. That protein is called tropomyosin. And then there's some small green proteins. That protein is called troponin. And the next slide that I show you shows the spelling of those terms. The thick filament is only made of one kind of protein called myosin. And it's a long protein that has these little heads that stick out. So when you put a bunch of these myosin molecules together, they have all these little heads that stick out. Now, it seems a little counterintuitive that the thin filament actually has 3 proteins associated with it, whereas the thick filament is only made of one kind of protein. They named these based on how they looked under the microscope. If you look at that microscope picture, you see some thin filaments and some thick filaments. So that's how they originally named thin filaments and thick filaments. But then once we did some biochemical studies and we could figure out what proteins make up these filaments and what their arrangements are, then that's how we determined that there's 3 types of proteins that are found in these thin filaments. So the thin filaments are made up of 3 proteins. The main one is called actin. So this is often sometimes called an actin filament. There's a long, thin protein. It's shown as the little green lines in that previous figure, and it's called tropomyosin. One important thing to tell you is that myosin and actin have a very high affinity for one another. They like to bind together. If at all possible, actin and myosin will bind together. And there are binding sites on those actin proteins for myosin. Tropomyosin blocks that or covers those binding sites. Because sometimes in muscle contraction you want that binding to take place, and then sometimes you don't. And so this is the protein that's going to control that. And we're going to see a lot more about that in just a little bit. Now, to hold tropomyosin in place is the third protein called troponin. It binds to actin and holds tropomyosin in place. And it's actually the ones that are the round green ones that are in that previous figure. Tropomyosin is more the long filamentous green protein that lies in the grooves of that actin helix. And then troponin is more of the round ones. Troponin kind of acts like a spot weld to hold tropomyosin in place. And the other thing about troponin that's kind of interesting that I'll tell you, that'll be significant a little bit later on, is that troponin is calcium sensitive. And that means that it can change its folding or change its conformation depending on whether or not calcium is bound to it. So that's a little bit of foreshadowing. We'll talk a little bit more about that when we talk about the calcium piece. Thick filaments are bundles of myosin proteins. And those myosin proteins have a kind of a long tail and a globular or rounded head region that we saw in that previous figure. The heads are what form the cross bridges to actin during contraction. And actin has a very strong affinity for myosin unless that interaction is blocked, and that interaction can be blocked by tropomyosin. So to give you a summary of the sliding filament mechanism of muscle contraction, the thin and thick filaments, the actin and myosin filaments, slide past each other to contract the muscle. The sarcomere shortens, which shortens the myofibril. The myosin head can change its shape or its folding in the process. So what happens is the myosin head binds to actin, and that binding is called a cross bridge. So the myosin head forms a cross bridge with actin. And that actin head can change its shape to slide the filaments relative to one another. ATP is required to break the cross bridge so that contraction can continue. And the way that we know that is people have studied the idea of rigor mortis, which is when the cells no longer produce ATP. After you're dead, your cells no longer make ATP. And if there's no ATP, those myosin cross bridges can't break. So there is a period of time after rest that the muscle contracts and cannot break that cross bridge between actin and myosin because there's no ATP around. And so that's the process that we sometimes refer to as rigor mortis. So I'm going to click on a link, and it's going to open a website for you, and that's going to show an animation about this sliding filament mechanism and the sarcomeres that we're talking about. So Windows should have opened up for you. This is an animation that comes with a different textbook. But if you look in that red bar, there's an area that says play. If you want to press that play button, now if you want to put your cursor on that little yellow button and slide it back to the beginning and actually keep your cursor on that button and just slide that button back and forth. All the way to the end, all the way to the beginning, all the way to the end, all the way to the beginning. That speeds up the process, and so you can actually see the shortening of that sarcomere that's going to form the basis of muscle contraction. So you can either minimize that window to get it out of your way, or you can close it to come back into the Wimba classroom. Sarah, you probably have a pop-up blocker on that blocks that because it pops up as a separate window. So what I can do is give me just a second to okay, it's not letting me paste anything into the chat window. All right, what I can do is do this. No, it's not letting me paste it there either. Probably what you're going to need to do is go back to the archive once the archive is posted and skip down to where it pushed that link out and do that without having your pop-up blocker on, and then it'll open for you, and then it'll pop up for you. I can also make sure and post the link. Actually, I believe it's already posted in the animations tab. Under the module tab, there's a series of animations. In the muscle contraction animations, it's one of the ones that's there. It's the sarcomere from McGraw-Hill that's there. So this is a figure from your book that's very similar, that shows some of the same processes going on. So it shows in this first portion the length of the sarcomere when the muscle is relaxed. And then in the second portion, it shows the length of the sarcomere when the muscle is contracted. And you can see that sarcomere shortens because those filaments slide relative to one another. So what they show you in part C, is they're showing you what's happening right before muscle contracts. And you can see this myosin head, you can see this actin filament. And it says when calcium gets released, and it's going to talk about that more in the next section, but when calcium gets released, a cross bridge can form. So now rather than there being a space between myosin and actin, now myosin is bound to actin. In part E, they show you these little arrows. And what they're showing you there is the myosin head kind of rotating or cocking. And when it does that, the two filaments slide relative to one another. And so in part F, it's showing you this sliding. Now, energy is involved in this process. ATP is required. New ATP has to bind in order to break that cross bridge. So if you don't have any ATP around, then you can't break that cross bridge. And so that's what your book is referring to when it's talking about the rigor mortis part. So in the next section, in section 6.4, your book shows that calcium ions are the key to muscle contraction. Calcium is the key to signaling to the thin and thick filaments of the muscle cell when it is time to contract. And it does that because that calcium binds to the troponin molecule. Remember, troponin held tropomyosin in place. When calcium binds to troponin, it changes its folding and it moves tropomyosin out of the way. That removes the block, so that's how actin can bind to myosin. So that's the link. Calcium is the link that starts this whole process. Calcium binds to troponin and removes the block so that that cross bridge can form and the steps can start to happen. Now, to understand a little bit more about these next few steps of the process, I want to talk to you a little bit about some of the structures in muscle cells. Now, these are some additional structures in addition to what we talked about before. These are all muscle cell structures that are in the muscle fiber or the muscle cell. So we're looking at the level of the muscle cell to look at these structures. There's a special name for the muscle cell membrane. It's called the sarcolemma. There's a special name for the muscle cell cytoplasm. It's called the sarcoplasm. There is a structure in the skeletal muscle cell. It's very similar to endoplasmic reticulum. Rather than being called endoplasmic reticulum, it's called sarcoplasmic reticulum. And so we often abbreviate it SR. It's a lot easier to say SR, and it's a lot easier to write SR. And that's an abbreviation we use for the sarcoplasmic reticulum. It's very similar to ER, and it stores calcium like ER does. We've already talked a little bit about the sarcomeres that contain thick and thin filaments. And then on the next figure, I'm going to show you these structures called T-tubules or transverse tubules. And these are kind of infoldings or invaginations of the sarcolemma, of the muscle cell membrane. And they're very close to the membrane of the SR. So these T-tubules are transverse tubules because they go transverse through the cell, are kind of infolding or invagination of the cell membrane. This allows the cell membrane to be able to really go deep into the cell. And the other thing that's important about it is it's very close to the membrane of the SR. Now, when we're talking about these membranes, these are all the same membranes that we learned about when we learned about cell structure in Chapter 3. So the muscle cell membrane is a phospholipid bilayer, just like all the other cell membranes that we've looked at, that has proteins in it that make it specialized. The SR and the T-tubules are all made of that same phospholipid bilayer with proteins in it. So it's the same type of we have been learning about with transport and the cell membrane. And this figure shows you some of these structures. They're showing you the plasma membrane, and they're showing you the T-tubules, which is a way for that plasma membrane to fold down into the cell. And then all of this yellow stuff that they're showing you inside the cell is the SR, which is all a membrane-bound organelle that stores calcium. So there's a nerve associated with each of these muscle cells. The endings of a motor neuron are close to these muscle cells. In fact, they show you in this picture, they show you the end of an axon of a motor neuron, and it's very close to this muscle cell. And it signals the release of calcium so that calcium can trigger the sliding filament mechanism in this sarcomere. So normally in the thin filament, tropomyosin blocks that cross bridge from forming. It blocks that binding, and it's held in place with troponin. And troponin is calcium sensitive. So when calcium gets released, its folding or its shape can change. So the release of calcium causes troponin to change its shape. And that moves tropomyosin out of the way and allows the cross bridge to form. And so this is the figure that shows that. Shows troponin holding tropomyosin in place and blocking the binding site. Then they tell you that a nerve causes calcium to be released, and they're showing calcium by these little blue balls. And so calcium binds to troponin and moves it out of the way. Now tropomyosin is moved out of the way, and these little dots or these myosin binding sites on actin are exposed. Troponin moves, twisting tropomyosin out of the way and uncovering those binding sites on actin. And now actin and myosin can form that cross bridge. And that sliding filament mechanism can take place. Now, when that nerve impulse stops, there's no more calcium. Troponin goes back to its original shape, the shape it has when there's no calcium around. And again, it covers the actin binding sites, and the cross bridge can't take place. So that has to happen in order for muscle to relax. So I want to go back to this figure just for a second, looking at part D and E, which shows us the structure of the thin filaments and the thick filaments. This was one of the figures that we started with when we talked about troponin, tropomyosin, and actin in the thin filament, and then myosin in the thick filament. And we said the way that muscle actually contracts, this figure showing the way that muscle actually contracts is those filaments slide relative to one another. That myosin head actually kind of walks its way down the actin filament or ratchets its way down the actin filament, drawing those actin filaments in and contracting that sarcomere. And if that happens in all the sarcomeres in the myofibril, then the myofibril contracts. If that happens in all the myofibrils in the muscle cell, then the muscle cell contracts. If that happens in all of the muscle cells in the muscle fascicle, then the muscle fascicle contracts. And if that happens in all the muscle fascicles in the muscle contracts. So we have to have a way to make all of it happen at the same time, and that way is by using calcium. So calcium signals to all the thin and thick filaments when it's time to contract. And the way calcium signals when it's time to contract is that calcium binds to troponin and moves tropomyosin out of the way so that the myosin head can bind and do that ratcheting and walking its way down that actin filament in order to contract. Now, we got a hint in this figure that nerves are involved. Nerves attached to muscle cells, and then they show you again here, a nerve attaches to a muscle cell. The area where a nerve and a muscle meet is called the neuromuscular junction. Neurons act on muscle cells at the neuromuscular junction. Nerve-muscle junction, where the nerve and the muscle meet, is called the neuromuscular junction. And so that nerve cell can send a signal to the muscle cell to cause that calcium to be released. So we're layering on a little bit more information here. So we know that a nerve signal causes calcium to be released. We saw that in the previous figure. Now we're going to talk about the details of how a nerve cell causes calcium to be released. There is always a space between a nerve and another cell, and that space is called a synapse. And so the electrical signal in that nerve causes a chemical to be released, and that chemical crosses the space. Those chemicals are called neurotransmitters. We're going to talk about neurotransmitters again when we get over to the nervous system. The specific neurotransmitter that's involved when we're talking about the neuromuscular junction, when we're talking about nerves getting muscles to contract, is one called acetylcholine, and it's often abbreviated ACH. So wherever you see this ACH, we're talking about acetylcholine. Acetylcholine is just a chemical that crosses the space between the nerve and the muscle cell. It can excite the muscle cell and cause it to contract because it causes the release of calcium from the SR. So here's a figure that shows you a blow-up of where this nerve comes down to this muscle cell. So they've really blown up where this yellow nerve meets this muscle cell. And so you can see there's a space called the synapse. In this ending of the axon, there are vesicles. And these vesicles contain neurotransmitters. And if we're trying to communicate with a muscle, that neurotransmitter is going to be acetylcholine. And then the muscle cell has receptors for acetylcholine. So let's think back a little bit to cell transport. Some things can get into the cell, and some things cannot get into the cell. It's based on the structure of the cell membrane. Small lipid-soluble molecules and gas and water can freely diffuse through the membrane. But most other molecules in the body, including neurotransmitters, have to have a protein. So here, these neurotransmitters are going to bind to these proteins that are in the cell membrane, these receptors. That tells the muscle cell that they're there. It's like an antenna on the top of the cell, and the antenna can detect the presence of these molecules, can pick up these molecules, because this molecule binds to this receptor. It says, hello, I'm here. And when that happens, that can send a signal through the whole muscle cell membrane, even down the T tubule. So it sends a signal through the whole muscle cell. And the T tubules are close to the SR. So when that signal goes through the whole muscle cell membrane, even through the T tubules, it hits the SR and causes it to be released from the SR. And I summarized that for you in this slide right here. So the nerve releases a neurotransmitter. It crosses that space between the cells. What's that space called? Synapse. It signals to the muscle cell to release calcium from the SR. And then that brings us to the piece that we just talked about. Calcium binds to troponin, moves tropomyosin out of the way. The cross bridge can form. And that brings us to the first piece that we talked about. Those protein filaments slide relative to one another, which shortens the sarcomeres, which shortens the myofibril, which shortens the muscle fiber, which shortens the muscle fascicle, which shortens the muscle, which moves bone. Now I have another, I'm going to click on this link, and this is going to push out another website to you that is a similar animation that's going to show you some of the events of the neuromuscular junction. So again, they're going to show you a nerve cell and a muscle cell. They're going to show you the vesicles. They're going to show you the events that happen there at the neuromuscular junction. So you can just press play on that to watch that. I always try to leave a little bit of time when I push a link out like that in case people want to actually go ahead and look at the link right then. If you don't want to look at it, if you don't want to press play and look at the animation right then when that window opens, you can just minimize it. Or you can come back into the archive and just skip to that link in the archive and open it and watch it at that point. But I always try to leave a little bit of time so if somebody wants to go ahead and look at it right then, they can. So that's what the silence was all about. So that animation has a lot of good details in it about this process, these events at the neuromuscular junction that are causing the nerve to stimulate the muscle to contract. And it has some things that we haven't mentioned yet, but once we go through some of the stuff in Chapter 13, If you come back and watch that animation, and I'll probably put it up again so that it'll open in the archive, you'll notice that some of the details we're filling in, even as we go through chapter 13, you'll see them in that video. So I really like that video because it kind of summarizes some of the things in figure 611, some of the things that we've got listed here, as well as some of the things that we'll talk about in chapter 13. Now, we can interfere with this process and therefore interfere with muscle contraction. So there are toxins that bacteria produce that cause paralysis by interfering with this process. So one of a very famous toxin that does that is from a Clostridium botulinum bacteria. It's called botulism toxin or botulinum toxin. It blocks the release of acetylcholine from those synaptic vesicles. That acetylcholine can never be released from the nerve, so the nerve thinks that it's stimulating the muscle to contract, and the muscle is never getting the acetylcholine. This bacteria can be found in improperly canned foods, and even a tiny amount of the toxin can cause death because it causes paralysis, even of the respiratory muscles. Now, we've figured out a way to use very small quantities of it in a very specific way as medicine. Some people have heard of it as Botox. It can be used to alleviate muscle contraction that causes crossed eyes or muscle contraction that causes uncontrollable blinking. It can relax muscle spasms, including vocal cord spasms. Most people have heard of it as a cosmetic treatment to relax muscles that cause facial wrinkles. And it pardon me, can also alleviate chronic pain due to muscle spasms that you have in the back or the legs. So this was originally one of the ones that I had in the list for the discussion board this week, and I've actually taken it out of the discussion board as an option because I wanted to use it as an example. So what I'm looking for in the discussion board post is that you give me the mechanism of how this is involved in muscle contraction. And so you're going to want to make sure and include details like this. All this other stuff is really interesting, but it doesn't answer the discussion board post. This is the piece that answers the discussion board post, because the discussion board post is asking about a mechanism. So a mechanism usually answers the question, how? How does something take place? How does this toxin cause paralysis? And usually a mechanism is going to have something to do with a process. If it's something to do with a discussion board post, then it's probably a process that we've studied. But if you're reading in the newspaper or they're talking about some sort of biological mechanism, they're talking about some sort of a process of some sort of steps that happen for something to happen. And so now that we've talked a little bit about the steps that happen for a nerve to stimulate a muscle, this is a good example of something that can stop that because it blocks the release of acetylcholine from those synaptic vesicles. So the acetylcholine never gets released. It never gets to the muscle cell. So there's nothing wrong with the muscle. This only blocks the transmission of the signal from the nerve to the muscle. So that's an example of one of the ways that you can answer the discussion board post this week. And just make sure that you're kind of telling me how this actually stops contraction. And we're going to do another example in just a second. So again, this is kind of a summary slide. The first part of the chapter talks about this piece, the structure of the myosin and actin filaments, and then the sliding filament theory. And then your book layered on the idea that calcium controls this process through troponin and tropomyosin. And then your book layered on, but this whole process is controlled by a nerve releasing a neurotransmitter. In this case, it's acetylcholine. So I wanted to put this summary slide in so that as you're studying each of these processes, that you kind of understand the whole sequence from beginning to end, and not just the pieces that they give to you, but that you can get kind of an overall view of what's going on too. So we've introduced the idea of nerves here because we're talking about how nerves stimulate muscles to contract. So to understand a little bit more about how neurons do that, we need to study a little bit more about the function of neurons. And so that's also why I wanted to include some of the stuff from Chapter 13 in this week also. So this is a picture of a nerve cell. It's very similar to some of the things we've already studied about cells in general. But if you look at the structure of a nerve cell, some of the things that are unique, it has this very long axon that ends in these axon terminals. Your book calls them axon endings, but a lot of books call them axon terminals. So you may see axon terminal used in some of the animations. Nerves receive and send information, and that's a one-way process. They receive information through these dendrites. They send information down the axon. It only goes one way down the axon, and then that information can be sent to other cells by releasing a neurotransmitter chemical from these axon endings, similar to what we've already talked about. So that's why they call this the input zone and the output zone. The input zone is going to be where information is received by this cell, and the output zone is going to be information where the information is sent to other cells by releasing a neurotransmitter at the synapse. Neurons and muscle cells, too, are considered to be excitable cells. They're excitable because they can carry electrical signals through their membrane. Nerve cells and muscle cells can do this, can carry electrical signals through their membrane. And what we mean by that is ions can flow across those membranes. And so that's what we mean by an electrical signal. And so why that happens has to do with the sodium-potassium pump. We talked about the sodium-potassium pump just a little when we talked about active transport, when we talked about how cells function, which was in Chapter 3. Page 56 talks a little bit about active transport and talks about the ways that we got things across the membrane and that idea of active transport that uses ATP. And there's some animations on the sodium-potassium pump too, back in Module 2. So make sure that you're not forgetting some of the previous information that we've gone over. Basically, the sodium-potassium pump, if we want to look at a membrane, phospholipid bilayer, we can look at a big protein that's in that phospholipid bilayer. The sodium-potassium pump is basically pumping 3 sodium ions outside the cell for every 2 potassium ions that it's bringing into the cell. So that kind of refreshes us a little bit about what the sodium-potassium pump does. And so every time the sodium-potassium pump works, it moves 3 sodium out and 2 potassium in. 3 sodium out and 2 potassium in. These are supposed to be pluses to denote positive ions. Or some of them do, some of them don't. But every time this transporter works, 3 of them go out and only 2 of them go in. So you can see after time, you build up a lot more positive charges outside the cell than inside the cell. So in addition to a concentration gradient, where you have a lot of sodium outside and a lot of potassium inside, it also creates an electrical gradient across the membrane. The cytoplasmic side is a little bit more negative compared to the outside because the outside has all of these extra positive charges. And that situation is called the resting transmembrane potential. It's set up by this sodium-potassium pump, and it's a form of potential energy. It has the potential to do physiological work, because it's similar to concentration gradients in that the charges are going to want to spread out, just like concentrations of molecules want to spread out. And so this is a figure from your book. This is figure 13.3. This is in chapter 13. So we're talking about the nervous system. We've skipped ahead a little bit. And so this is showing you the sodium-potassium pump. Sodium-potassium pump sets up all of these conditions. Active transporters pump sodium and potassium across the membrane against their concentration gradient. And so we build up a high concentration of sodium outside the cell, a high concentration of potassium inside the cell, and a little bit of negative charges along this inner membrane. And that's the resting transmembrane potential. And that's the way nerves are all the time when they're not signaling. That's why it's called the resting transmembrane potential. That's a very normal situation for nerves to be in. Now, the other interesting thing is that muscle cells also have a lot of sodium-potassium pumps in their membrane. Muscle cells also have a resting transmembrane potential. Muscle cells are also like this when they're at rest, when they are not contracting. So another concept to review is the idea of ion channels. Some ion channels are what we call gated channels, meaning they can be closed and opened rather than just being open all the time. Things that can cause gated ion channels to open are things like a change in membrane voltage, voltage-gated channels, and the presence of a chemical. Those are ligand-gated channels or chemically gated channels. And they can be sensitive to different chemicals, like, for example, acetylcholine. There's also mechanically gated channels that respond to pressure and things like that. For our purposes, for our talking about the action potential, it's basically voltage-gated channels and chemically gated channels that we're going to focus on. Nerve impulses are often called action potentials, but an action potential is just this electrical signal that goes through the membrane. So actually, a muscle cell can conduct an action potential also. Any cell with this resting transmembrane potential that has these gated ion channels in it can conduct an action potential. So it can happen in a nerve going down the axon, and it can happen in a muscle going down the T tubules and hitting the SR to cause calcium to be released. So something opens a sodium channel, and sodium goes down its concentration gradient to enter the cell. This electrical disturbance, now we have positive charges coming into the cell when we used to have a lot of positive charges outside the cell in the resting transmembrane potential. There's a lot of positive charges outside the cell. Now when we open a sodium channel, and sodium starts to flow down its concentration gradient, that changes the voltage. It can open other voltage-gated channels if it reaches threshold. So here is a series of figures from your book. They're showing you the outside of the cell, and this orange area is the inside of the cell. In a membrane at rest, the inside is a little bit negative relative to the outside. You have lots of these positive charges outside the cell compared to inside. If you have enough sodium ions that come into the cell, it changes the voltage and causes more voltage-gated channels to open. This continues that same figure. The voltage-gated channels, like voltage-gated potassium channels, allow potassium to leave the cell. And that actually happens in series all the way down the membrane. So if we draw just a stretch of membrane, so this is part of the axon. So the cell body is this way, and the axon terminal is this way. And outside the cell is here, and inside the cell is here. So we have a little bit negative, and we have these channels in series. And they go all the way down this axon. 2, 3, 4, 5, 6, 7, 8, 9, 10. And I'm going to attempt to do colors here. So let's do, so this is a sodium channel. And then we'll do blue for potassium. And they are interspersed with potassium channels. They alternate going all the way down the membrane, similar to what was shown to you in that previous figure. And so something opens this first sodium channel, and sodium flows down its concentration gradient into the cell. And because it's a positive ion and it's coming back into the cell, that disrupts the membrane voltage. It's no longer at that resting transmembrane potential. And if it changes the voltage enough, the next channel in the series, the membrane voltage has been disrupted, so that opens a voltage-gated channel, and potassium can flow down its concentration gradient. Now that's taking those positive ions back out of the cell, so that changes the membrane voltage. If it does it enough, it opens the next voltage-gated channel, which is sodium, and sodium flows in and disrupts the voltage enough to open the next channel, and potassium goes out, which opens the next channel, and sodium comes in, which opens the next channel, and potassium goes out, which opens the next channel, and sodium comes in. Kind of regretting doing quite that many, but you get the idea. So movement of ions through one channel changes the voltage and opens the next channel. The movement of ions through that channel changes the voltage and opens the next channel. It's very similar to setting up dominoes and pushing down the first domino and having that cascading effect where each of the dominoes hits the next one and they all fall down in a line. It's very similar to that. We say the action potential is an all or nothing, because if you reach threshold on this first one, if you push that domino down hard enough, then everything else will just flow all the rest of the way down the axon. Now, the very last channel in series is a voltage-gated calcium channel. So when we get all the way down to this voltage-gated calcium channel, calcium comes in, and calcium triggers exocytosis of our vesicles that contain neurotransmitter, so that that neurotransmitter can signal to the next cell what it's supposed to do. And if that next cell is a muscle cell, that starts the events that we've already talked about in the muscle cell. And so that's detailed for you in this figure, 13.7, that's in your book on page 246. So when that action potential gets all the way down to that axon terminal, the axon terminal has those, hang on, circled the wrong thing. The axon terminal stores those neurotransmitters in vesicles. And when that action potential gets to that axon terminal, it causes exocytosis. We already know that piece from the previous chapter that we studied. We already know that piece from chapter 6 on the muscle. They show you here the plasma membrane of the muscle cell has receptors for the neurotransmitter. They're giving you an additional little bit of information here. That receptor is a sodium channel. Binding of the neurotransmitter to that sodium channel opens it up, and then ions can flow through that sodium channel. So this is a chemically gated sodium channel in the muscle cell membrane. But what's happening is sodium flows into the cell, which is the first step of an action potential. And so that starts an action potential in the muscle cell also. So we can go back and we can tie this together with what we were talking about the muscle. The whole process starts with the events of the neuromuscular junction. So now we can add to that. The action potential arrives at the axon terminal from that previous figure. Calcium enters the axon terminal and initiates exocytosis, which is the part that releases the neurotransmitter, crosses the space between the cells. And now we have a lot more detail from that last figure, from figure 13.7. We have a lot more information on that. Crosses the space between the cells, binds to a receptor on the muscle cell. What is that receptor? That receptor is a ligand-gated or chemically gated sodium channel. So when it opens, sodium comes into the muscle cell, and sodium starts an action potential. That action potential goes all the way down. That's what we mean by signal. That action potential goes all the way down through the whole muscle cell membrane, even down the T-tubules. It hits the SR and causes calcium to be released from the SR. Then calcium binds to troponin, moves tropomyosin out of the way, forms the cross bridge. They slide, shortens the sarcomeres, causes muscle contraction. So that's one of the reasons why I wanted to fit these two chapters together, because they really go hand in hand. And we can study chapter 6 and then we can wait a couple weeks and study chapter 13. But I think to put them both together like this because they both relate to this process. And some of the things that are in chapter 13 help us to understand some of the things in chapter 6. Like this. This is talking about the synaptic cleft. This is talking about the muscle cell has these receptors for the neurotransmitter. Tells you now that it's an ion channel, and when it opens, it allows sodium to flow, and that starts an action potential in the muscle cell membrane. So I think they fit together really, really well. Now here's another toxin that we can talk about how it interferes with the process. If we can learn how things interfere with the process, that helps us to learn the process. And so here's another one, curare. Curare comes from a plant. It's a poison that's often used by South American Indians on their blow dart arrows to try to paralyze prey, to try to paralyze animals that they're trying to kill. It causes muscle paralysis. How? Again, if you're talking about a mechanism, you're going to want to be answering the question, how? Blocks muscle paralysis. How? It blocks the acetylcholine receptors, inhibiting those sodium ion channels. So now that we know from figure 13.7, now that we know this piece, this binding of the neurotransmitter that opens and the ions flow in, and that's what starts the signal to the muscle cell, it makes sense that if you block this, you couldn't start the action potential that causes calcium to be released. And calcium is what triggers all of the other steps. And so curare blocks this particular step, and that's how it paralyzes prey. The Botox blocks this step. So the step of exocytosis is what's blocked by Botox. The step of the muscle cell receiving the information is what's blocked by curare. And there's a number of toxins that block a lot of these processes. There are some toxins that block the sodium channel in the action potential, and that's how they work. There are some that block potassium the action potential, and that's how they work. So make sure when you're working on your discussion board post this week that you're trying to include an answer to the question, how? How does this cause paralysis? How does this affect the muscle cell, or how does this affect the nerve cell? And I think if you do that, then you'll be in good shape as far as putting your answer together. Okay, I knew I would go over. And I wanted to make sure that I went through both of those kind of together because I think that they fit together pretty well. So if there's any questions that folks have, please feel free to email me or post them to the discussion board. And I'm hoping that going through some of those examples helped people think about how you want to do your discussion board post this week. I haven't been into the discussion board, but if anybody has already done a post to the discussion board, but this discussion helped to clarify anything, please feel free to go back and reply to yourself and add anything that you want to, and that can be included with your discussion board post when I go to grade it next week. So I'm hoping that that helps clear up some of the questions that you might have had from the material, and just let me know if you guys have any other questions.